Hello everybody and welcome back. So today we will talk about polymerase chain reaction, in short PCR. So this is a technique that will allow us to make more copies of DNA. In other words, this is a technique of amplification of DNA. So we have seen how the DNA replication happens inside the cells, in eukaryotic cells, that it involves so many number of enzymes, and it's a stepwise reactions, quite complicated. Many enzymes are involved. Along with that, some ingredients are also needed. So polymerase chain reaction is a method that will allow us to do the DNA amplification in laboratory, in vitro. So when something happens inside the living organisms, living cells, we call it in vivo. Outside the living cells, for example, in the laboratory that you are mimicking, is called in vitro. So we will see today how you can do the amplification of DNA, how you can make more copies of DNA in your laboratory. And this is absolutely essential thing, because the amount of DNA that you can isolate from different sources during your studies, for example diseased cells, for example microorganisms, you have isolated DNA from the microorganisms, from diseased cells and other sources, blood cells and so on, and you want to study the various properties of those genes. Then the crucial thing is that the amount that you isolate is actually very, very small, and with that you cannot do your laboratory stuffs, you cannot do many experiments with them. So it's absolutely essential that we need more quantity of those genes or more quantity of those DNA. Let's say somebody, you have seen, we collect the people or the scientists, or even for the forensic teams, they collect hair samples or the blood samples, skin samples from diseased persons. If there are many casualties, some mishap happens and there are many casualties, you are not identify those persons. So many times what people do nowadays, the forensic team, they collect even the hair samples and then isolate the DNA, and then they find out the identity of the person. So you can imagine the amount of DNA that you can isolate from a hair sample is so tiny, so small, that with that you cannot carry out proper investigations in the laboratory. So you really need a large more quantity of material, more quantity of DNA to carry out your needs, to carry out your experiments in the laboratory. So it is absolutely essential to increase the quantity of the DNA, and this is the technique that will allow you to make more quantities of the gene or the DNA that you have isolated. That is the beauty of this method, and that is where comes the importance of this method. So until PCR technique, the biochemistry or the biochemical techniques, even some chemistry tools, were stuck. So after the invention of PCR, it has given a huge boost to the biochemist people, to the biotechnologists, and even to the chemists. That has really increased the level of the research. So the key person who has figured out all the real steps for the PCR, polymerase chain reaction, is Kary Mullis. Kary Mullis is the person who has developed the whole systems of the PCR. The PCR technique was actually fully designed or fully developed back in 1983, quite recently actually, not very long, and he received Nobel Prize in 1993, and this is one of the really biggest inventions in in the world or in the research of biochemistry, biotechnology and chemistry. You know, Kary-Mullis has actually received the Nobel Prize in chemistry, not in physiology and medicine, because as we will see later very soon, all the methods that you are going to use in the laboratory, they involve chemical processes, chemical reactions. So that is what is the background of the polymerase chain reactions. As the name suggests, of course, you can understand it needs a polymerase, and it's a process or it's a method that is based on the enzyme polymerase. So PCR is a technique to amplify genomic DNA that we have extracted from organisms. It's one of the most important discoveries that has revolutionized biochemical or biological research. It's very, very true actually. What are the applications? Identifying the cDNA means the chromosomal DNA or any genomic DNA. Detection of genomic mutations, that is one of the key reasons that we need to study the genes, because if you isolate a gene from a diseased cells, we need to know whether there are mutations inside it, or whether they are the same gene as the normal, or what kind of abnormality has happened to those genes. So genomic mutations, diagnosis of a genetic diseases. So there are many changes, alterations that happens in the genome. Because of that, the disease like Parkinson's, disease like diabetes, disease like cancers happen. So for disease diagnosis, this is an absolutely important tool. The principle we will shortly see. It's a complementary to DNA replication. Now, if you recall the DNA replication process that has been happening inside the cell, so we have seen it goes through multiple steps. Now the question is, if you want to do the same sort of steps, can you do the same process in your laboratory? Can you do DNA amplification or the DNA replication that we have seen in a test tube? So let's see that and then assess whether the same process can be mimicked in your laboratory, and if not, what are the problems and how can you solve them. So first, if you remember, for DNA replication process, what are the ingredients that have been that were needed? DNA replication, that is in cell, in living organisms. Ingredients, and here I will write the enzymes. What are the reagents that were required and what are the enzymes that were required? What are the reagents or the ingredients that were required? Of course, in the middle of it, you needed the sample DNA, the DNA that you want to replicate, or your parent DNA. Or gene. That is what you require first, because that is what you want to make more copies of. Second, what you need? You needed RNA primers, if I remember correctly. So first, you needed actually multiple number of RNA primers, because if you remember, the leading strand required one RNA primer, while the lagging strand required many number of RNA primers, because you were starting from the middle of the strand, from somewhere in between region of the strands. So every time the strand was opening up, you had to start with a new RNA primer. So many number of RNA primers were required. What else did you need? You needed the DNTPs, the free nucleic acids. 4 DNTPs were required, of course, for example, DATP, adenosine triphosphate. You needed DGTP, guanosine triphosphate. you needed GCTP, cytidine triphosphate, and then you needed DTTP, thymidine triphosphate. So, general formula being this. This is the base A, T, G, C. The structure was this: 3' hydroxyl free O and the phosphate, triphosphate. In the 5' end. This is what you needed. Did you need anything else? RNA primers, DNTPs? Mostly those ingredients were needed, and you needed a lot of enzymes. The first enzyme that you required was helicase. Then you needed single stranded binding protein. After that came the polymerases. You needed 2 polymerases actually for 2 strands, if you remembered. Before that, you of course needed a RNA primase that synthesized the RNA primers. So, RNA primase was needed. After that, you needed RNase to destroy all the RNA primers. Then you needed DNA ligase to stitch all the fragmented DNAs that you have formed. So, at least these are the number of enzymes that you required, and these are the ingredients, chemical structures that you required for the replication of DNA inside the cell. Now, the question is, if you take all of these together in a test tube, are you going to be successful? Is it going to work in doing the replication? So, if we see, let us say this is your test tube, and you put first your double stranded DNA, target DNA. This is your double stranded DNA, 5' to 3', 3' to 5'. All the hydrogen bondings are there. Now, the first thing was you had to open the double helix, and you needed the helicase. So, you had helicase here that is going to open up. Make a replication fork, and then that had to be stabilized by SSB protein. So far so good. Now, what you need? Now, you need the RNA primers. Okay, add the RNA primers. Maybe with the help of the primase enzyme, and that will start the replication. So, this is your RNA. That was to be 5'. You have 3' hydroxyl group free. Here the primer would be this with the 3' hydroxyl group free. Good enough. Now, what you need? You need to replicate, you need the extension, you need to synthesize the complementary strand that is done by polymerase. So, you add the 2 polymerases for 2 different strands. So, the polymerase is going to bind here, and then you need the all the 4 DNTPs. So, suitably according to what you have on the opposite strand, that the complementary triphosphate would be taken up by the polymerase and it will go on continuing the synthesis. Same thing will happen here. So, you will get so this is RNA and you will synthesize the DNA on it. So, this is your DNA, this is your DNA. So, your half portion is finished and then you can open up more and can go back. That is how you will get the leading strand. We have seen 5 prime to 3 prime, that is the leading strand, and you can synthesize continuously the complementary new strand, that is 5 prime to 3 prime, this here constant, plus this is your lagging strand, parent 3 prime to 5 prime, and you will synthesize the DNA in fragments. All the gap were originally occupied by the RNA primers that you have digested, that you have to cleave by the enzyme RNase, and then you have used ligase, DNA ligase, to get the full complementary strand, 3 prime to 5 prime, 5 prime to 3 prime for this. So, from one DNA you can synthesize in the first cycle 2 DNAs, fine. Now, this thing should go on and on in a cycle because you have added all the enzymes, they are not going to stop there, and you need more quantities of DNA. So, now think of the next cycle, is it going to work? Is this and this going to be replicated further under your condition in your test tube? I do not think so. I do not think it is going to work in the next cycle anymore. Once you have added all the ingredients in the same test tube, then let us see whether it can go on. First thing is that you have added RNAs there in your test tube. So, for the second cycle also you need RNA primers for this strand and this strand, all strands basically you need RNA primers. So, even if you add second cycle, five prime 3 prime, you had opened the second replication fork for one strand, 3 prime 5 prime, you need the RNA primer here, you need the RNA primer there. This is your test tube that contains all of them already, but you had RNAs there. So, RNase has actually cleaved off or destroyed all the RNA that you have used before. So, even if you add new RNA primers, are they going to work? No, because you already have the RNAs present in your test tube. The moment you add the RNA primers, they will be destroyed. So, all the RNA would be destroyed, and if the RNAs are not there, the extensions cannot happen, polymerase cannot react. That is problem number 1. Second problem is, you see, you have synthesized one new strand, two new strands there. So, you have some copies of DNA already in your solution. You can write it this way, that you have a double stranded DNA, 5 prime to 3 prime, 3 prime to 5 prime, and here you have again some copies may be the same, 5 prime 3 prime, 3 prime 5 prime, some copies are floating around in your solution, and you have DNA ligase there. So, the very first thing what will happen is DNA ligase will stitch together whatever DNA you have in the test tube. So, these two would be stitched together, these two would be stitched together, and instead of getting your real DNA, you will get now longer version of DNA. So, your originality is lost. Moreover, if you think that even if it goes without the RNAs, even it can synthesize the next strands, you are going to synthesize the fragmented DNAs, and those fragmented DNAs, before they are synthesized, I mean right after they are synthesized, right after they have been made, they can non-specifically connect to each other because you have ligase present already in your solution. So, several problems. In other words, if you want to do using the same sort of same set of enzymes, using the same set of ingredients that happens in a biological cell, you are not going to get your amplified DNA. The process of DNA replication cannot be performed smoothly in a test tube without the living cells. So, that is the problem of mimicking the polymerase, mimicking the DNA replication in the lab. So, what is the way out? How can you change this? How can you make it work? So, one thing you have to remember is that you are in a laboratory and you are doing it in a test tube. So, you have some freedom that you would not have in a living cell. So, we have to think that way and think what we can do to develop or to find out a new method to so that the process may work. So, if you look, you have initially your target DNA, double stranded DNA. What was the first criteria? The first criteria was that you need to unfold the DNA, at least partially. That is why you needed the enzyme helicase, and that is why you needed the enzyme single stranded binding protein. Now think of it, can you do this thing in your laboratory? Can you unfold your DNA or the gene in your laboratory, or what should be the condition if you want to open them up? You remember we have talked about DNA denaturation. DNA denaturation means that to a double stranded DNA would be split into two single stranded DNA, that is DNA denaturation, right. So, now think of a way how can you do a DNA denaturations. So, basically means that you have to break all the non-covalent interactions that is present within your DNA. All the hydrogen bonds, all the pi stacking interactions should be broken down. And in your laboratory, what is the tool you have to do it? Very simple, very simple. By heat. If you simply heat it up to a little bit higher temperature, we usually do it at 95 degrees Celsius. You do not go to 100 degrees Celsius because in that temperature your solution, which usually in water, DNA is soluble, very much soluble in water. So, most of the work of DNA we do in aqueous medium. There are other buffers in it, but the medium is aqueous. So, it will start boiling at close to 100 degrees Celsius temperature, and so we do not want that. That is why the temperature is kept little bit less, close to 95 degrees Celsius. If you heat your DNA at 95 degrees Celsius, what is going to happen? It is going to break all your non-covalent interactions. So, at this stage, will be broken, which includes hydrogen bondings, pi stacking and all. So, in other words, your base pairs would be destroyed. and you can very much have two single stranded DNA free from each other, no interaction at all. So, by simple trick of heat, you can separate the DNA into two single stranded DNA. Now, of course, you can understand the same condition obviously cannot be used in cellular system, because 95 degree Celsius temperature inside our cells, if you want to increase that temperature, we will all die. So, that is what the freedom we have in the laboratory. Then you can use heat to make the double stranded DNA into the single stranded DNA. What is the other advantage? In cell, you have opened the DNA or you have opened the gene partially, and that is where all the problems were in the lagging strand. Here you see, once you put heat, they are completely separated. Whole of the double strands is completely separated, not partial separation, complete separation of the double strand into two individual single strands. So, that gives you full freedom of the single strands, and now you do not have the replication fork, they are all open, and as we will see, you can understand that it makes the process so simple now, because of this simple treatment, because of this very simple ingredient, that is the heat in this case. So, since you have opened with the heat, you do not need helicase, it is not needed. Your SSB protein not needed. Now, you move on to the next step. What else is required? So, let us take this strand first. I still call it leading strand and lagging strand, which was originally being called in the cell, but they are no longer actually leading strand, they are no longer lagging strand, they will behave as equal. Now, we will see. So, if you take this 5 prime to 3 prime, now if you want to make the complementary strand, what you need? For cell, you needed the RNA primers that were synthesized from the RNA primase. Now, in laboratory, you do not need RNA primers, because we have already learned to synthesize oligonucleotides. So, we will use DNA primers instead of the RNA primers. So, now we will use DNA primer. In this case, this is only one. So, DNA primer. RNA primer is not required for the laboratory technique, because the DNA primers do not exist in our body. The short sequences, short oligonucleotide sequences of DNA do not present in our body. RNA oligonucleotides are present to some extent. So, since you do not require the RNA primers, therefore, RNA primase, that enzyme, is also not required. It is not required. Therefore, RNA primers are also not required. So, you see how the process becomes simplified one by one. So, you use a DNA primer, and now for the extension, of course, you need a polymerase, that is needed, because the reaction that I have shown, that I am not writing the whole thing again. Here OPPP triphosphate, this 3 prime hydroxyl which is here. So, this is 5 prime, this is 3 prime, will react with B2, this is OPPP. This reaction can be catalyzed, or this reaction can be done only by this enzyme polymerase. there is no other go. So, we need the enzyme polymerase, and you need all the individual DNTPs, that is also required. So, since DNA primer is there, DNA polymerase is going there bound, and suitably the complementary DNTP will be picked up and your strand would be synthesized in this direction, 5 prime to 3 prime. So, this is the new strand that you are synthesizing, that you have synthesized, 5 prime to 3 prime, continuous without a break or without any interruption. Now, since you did not use RNA primer, here all DNA, so you need not go any other steps. Your amplification for this strand is complete. Now, look at the other strand. Other strand was 5 prime. So, this is 5 prime to 3 prime, this is 3 prime to 5 prime. I will write here so that it will be more clear, 3 prime to 5 prime, that is this strand, lagging strand, 3 prime to 5 prime. Now, that is also free fully. So, in lagging strand, in DNA replication in cell, you have seen that RNA primer came and was bound in the middle of somewhere, because you were half open there or partially open there. In this case, your strand is fully open. So, and your reaction is always starting from the 5 prime to 3 prime direction. What is the 5 prime to 3 prime direction? Here, 5 prime to 3 prime. So, you are not starting at the middle of somewhere, you are starting at the very end, just like the leading strand. So, you are again, you can use another DNA primer, suitable DNA primer that is suitable for this strand, and then of course use the same enzyme. Now you need a polymerase, but do you require a different polymerase? No. Here is the same thing, 5 prime to 3 prime, 3 prime hydroxyl is free. For lagging strand, since your synthesis was happening in discrete manner in the middle part or somewhere in the portion of the gene, you had required a different polymerase. In this case, it is the same reaction is happening exactly same as here. So, the same polymerase will do it. You do not need a different polymerase. So, the same polymerase that you have used in the earlier stage will also work here, and the same DNTPs that you have in your solution will be used, and you can have a continuous synthesis without any interruption. So, this is your parent strand 3 prime to 5 prime, and this is what the new strand that you are synthesizing, 5 prime to 3 prime. So, you see both leading strand and the lagging strand are behaving as equal in a test tube. And since this is happening, you are not creating the Okazaki fragments, and therefore, you do not need the DNA ligase to ligate them, that is not required. And since you do not have the RNA primers, you do not need to destroy them. So, the enzyme RNase is also not required. So, what you do need here is, if I now go back and check what I had written initially here and there, you do not need for the laboratory one, you do not need the RNA primers, instead you need only DNA primer, and you need 2 of them, 1 for each strand. RNA primers were needed in multiple numbers. DNTPs, yes. These 2 are needed. Enzymes, helicase, no need. Single stranded binding protein, no need. polymerase you need or one. RNA primers no need, RNAs no need, DNA ligase you do not need. So, you have come down to a single enzyme, that is polymerase, for the whole process, and a single trick of heat, and the ingredient, one extra ingredient, that is the DNA primer, which you can synthesize. So, that is the beauty of doing this replication process in the test tube. So, now basically what you can have your test tube, you can take all the DNTPs 4, you can use a single polymerase, and you need the DNA primers, 2 DNA primers, and you will have your amplification as you move on. So, that is the brilliant development of this method, that you have simplified such a complicated process into a really doable thing. Now, what another requirement you have to use heat here, right, and then only you will get your amplified DNA, more number of DNAs, number of copies of DNA you are going to get. Now, there is still a problem. Do you see the problem? If you use all the 4 DNTPs, if you use the DNA primers, if you use the polymerase, and you have your target DNA, double stranded DNA, actually still you are not going to get your amplified stuffs. Why? There is a key problem. The problem is you are using heat close to 95 degrees Celsius, and in this temperature obviously we are breaking the DNA, we are denaturing the DNA. At the same time you will denature the enzyme also. The polymerase will be denatured at high temperature. So, polymerase is the protein, and the protein functions when it is in 3 tertiary structures, coil form, 3-dimensional form. I will show you the structure of polymerase briefly in a just very soon. So, but any protein when it functions, enzymes, they are all in a coil form, folded conformation, and those conformations are formed along with some covalent interactions, mostly the non-covalent interactions, hydrogen bonding, electrostatic interactions, hydrophobic interactions. At high temperature those will all be gone and your protein will be dead. Polymerase will be as good as inactive. So, at that temperature your polymerase cannot work, and that was a key problem that had been sustained for a long time, and people were searching to find a solution. What can be done? Can this be achieved at all? So, finally, and this is again another very important discovery, is that they have isolated one kind of polymerase that is known as Taq polymerase. This is a polymerase that can sustain high temperature. Because this polymerase was isolated from the microorganisms present in hot spring. So, hot springs have high temperature, close to boiling temperature, but still there are many microorganisms living there, thermophilic bacteria we call them, or thermostatic microorganisms, thermophilic microorganisms. There are life still in the hot springs. So, what they have done, they have, and of course, since there is a life, they were having their cell divisions as well. So, they must have polymerase. We have isolated those polymerases, and that is known as Taq polymerase. I will show you why Taq. So, the Taq polymerase can sustain high temperature, and now your problem is solved. So, if you now use this in your laboratory with the DNTPs, with the DNA primers, and the newly found polymerase, high temperature, you are going to get your amplification of DNA. So, finding out Taq polymerase was a crucial part of this whole method development. So, now I will show you how is it done. So, the whole of the PCR is done inside a machine. It is a very small machine, bench top, you can put it on your table, small machine which can be programmed. So, you can see the buttons here, it can be fully programmed. All the steps it can be pre-programmed so that it will go on automatically. Here is a sample chamber where the things are done, and typically you use a very small quantity of volume, less than 200 microliter most of the times, less than 200 microliter. So, the test tubes I was talking about are actually the PCR tubes, it is this small of size, very quite small size. So, everything is done here, and the key steps or the key things that you need here is, of course, you need the DNA sample that you have isolated, and that is going to act as your template. Second is you need the polymerase, Taq polymerase. Number 3, you need the oligonucleotides, which we call primers. So, 2 DNA primers, 4 is all the 4 DNTPs, DATP, DGTP, DCTP and DTTP. These are the 4 DNTPs you need. And lastly, you remember when we make a DNA hybridization, we always had to use salt, because phosphates that are present in the backbone will repel to each other. So, in order to overcome that, we always use either sodium chloride or magnesium chloride. And the second reason you need to use a magnesium chloride is because the polymerase uses magnesium chloride as its cofactor. So, a fifth thing is you need magnesium chloride buffer solution. So, next we will see how the steps actually work. Thank you.